Howdy, my name is Tate McCrell. For my research project, I did insect olfaction of macronutrients. I used macronutrient food sources to test the rate of insect olfaction, um, as demonstrated by the number of insects which were caught in a nearby light trap, um, which was proximal to the bait, the macronutrient baits, in order to detect how many insects each bait drew to the light trap. My research question was which macronutrients are detected by the most flying insects at night. And I decided to answer this question by running an experiment, placing different macronutrient fuel sources uh, near a UV bucket trap and then counting the insects the following morning. My hypothesis was that sugar would be the most appealing macronutrient for flying insects. The logic behind this is that um, sugar is a dietary staple for so many common flying insects, such as flies, as well as pollinators like moths or butterflies or bees. Um, and also just on account of the fact that sugar is such a fundamental component to most organic energy cycles. For my supporting literature, I have 4 articles that I looked at, which fed into my reflection on my research project. The first of which is The Many Roles of Fats in Overwintering Insects, which was a very interesting article which reflected on the role of lipid consumption among insects, specifically insects that overwinter, on account of the fact that this research project was run in October, and it was run in Iowa, where we have somewhat severe winters. I thought that this specific article was very important, and it was important to reflect on uh, the role of lipids in insect diets, in the diets of insects that uh, overwinter, as well as the fact that some insects do not digest lipids or do not have lipids as a significant part of their diet. The second piece of literature I looked at was the genomics of adaptation in host plants, to host plants in herbivorous insects. And the key component to this, which I took back to my research, was the concept of adaptations by the host plants, which would lead insects away from detecting macronutrients themselves through olfaction, and the idea that they would prefer to detect their preferred food sources, um, and detect other indicators of resource and nutrients than just the macronutrients themselves. The fourth article I looked at was The Evolution of Insect Olfaction, and this article uh, desc- described the development of insect olfaction and what we can ascertain about how insects developed the sense of smell. Um, and the and olfaction is and nutrient finding is one of the most key components to why insects develop olfaction, um, and that is explored within this article, as well as exploring physiological differences between insects. My fourth article that I looked at was called "Towards an Understanding of the Structural Basics for Insect Olfaction by Odorant Receptors," and this is sort of similar to the last one. It examined the physical structure of insect olfactory organs significantly, and I think that the most important question between the two last articles uh, is whether or not insects who are capable of feeding on macronutrient sources detect macronutrient sources directly via olfaction. For my methods, uh, I used a handful of materials here. I used a UV insect trap, such as the one shown on the right. I used an extension cord. I used bowls to hold my macronutrient baits, and then I used the macronutrient baits themselves. For my macronutrient baits, I selected butter to be a source of lipids, sugar water to be a source of carbohydrates, and turkey to be a source of protein. I thought these were sufficiently concentrated sources of each of these macronutrients while maintaining a certain organic quality that I thought might be attractive to insects, and yeah. For my methods, I've firstly scouted a location to set up the UV trap, uh, and that location was selected to avoid confounding olfactory signals and other confounding signals which might affect my results. I then set the UV trap up using the extension cord, and I for each night I would set the bait in a bowl and set that bowl on top of the insect trap according to a bait schedule. And the bait schedule is the nightly schedule for which bait is to be set on the UV trap. I ran 2 sets of trials such that there was a rotation which went lipid, carbohydrate, protein. So the first night would be lipid, etc. And then there was a skip day in the middle, and then I did it again. Baits are set at sundown and removed at 8 a.m. At 8 a.m., the contents of the insect trap were also removed and placed in a separate container to be counted, and the UV light is turned off. At sundown, the UV light is turned back on, and the next bait, or nothing, if it's the skip day, is set up. After 2 rotations, I had my data, and it was ready to be analyzed. Here are my results. As you can see in the table in the top left, that's my raw data. On the top right, I did a t-test comparison between each of the 3 categories. So carb versus lipid, lipid protein, and protein carb, in order to determine if there was a significant difference between any two of the categories. And as you can see with the lipid protein, that returns a t-test score of 0, which means it is statistically the results are statistically insignificant implicitly because a t-test can't really be run. The null hypothesis for my research project would be that there was no statistical difference between the number of insects collected for each macronutrient bait. My acting hypothesis was that there would be a significant difference between any 2 baits, and that sugar would be the most would receive the most insects. I used 3 different t-tests in order to establish whether or not there was a significant difference between any 2 of the baits. Using these, we established a t-score for each pairing. On account of the fact that lipid and protein results are the same, the t-test results are inconclusive, as I said earlier. The p-values representing carb protein and carb lipid are 0.3818, which puts our alpha above the uncertainty level, which makes my results statistically insignificant. So on account of that, and on account of the fact that the lipid and protein results are the same, my results are on the whole statistically insignificant. Going into this, I thought that there would be more insects each morning, and I thought that would beef up my numbers so that statistical significance would have been within the realm of possibility. Were I to do this research project, I would have more trials. And then for my confounding variables, it is worth noting that the week of testing just so happened to be unseasonably cold, and I think that may have had a significant influence on the results, as well as the fact that insect populations vary throughout the year, and that were this test to be run in July, I think I would have caught much more insects. Were I to run this test again as well, I contemplated. In reflection, I sort of thought that perhaps a sticky trap, maybe a custom sticky trap that would also emit olfactory signals for these macronutrients, might have more significant results on account of the fact that for this test, insects had to be drawn by the bait and then also drawn by the UV trap afterwards in order for me to collect them as samples. Here is my bibliography. These are the citations for the previously acknowledged literature. Thank you so much for viewing my entomology project. Have a good winter. Have a good Christmas.